Pfizer representative on the FRMA, Patient-Focused Drug Development Working Group, and the Patient Engagement Pillar of the Clinical Research Expert Group at the FPIA. Brett is the Deputy Topic Lead representing the ICH E22 Expert Working Group for general consideration for patient preference studies, and this is the industry lead for the Innovative Health Initiative Public-Private Partnership Project for patient-centered clinical study endpoints derived using digital health technologies. So please, thank you.
Thank you for the introduction, and thank you for inviting me to join you. I have two disclaimers other than my usual disclaimers. One, I am not European, and B, I'm not a statistician. So I appreciate you allowing me to join you here. I am, however, an economist by training, so I do know a little bit of econometrics, enough to do what I do, but I only know how to do one thing, and that's what I'll show you here today. Full disclosure, I am speaking on behalf of myself and not on behalf of Pfizer or anybody else that's mentioned in this presentation. I'll take you through this case study, which involves a submission for marketing authorization that we made to CHMP for a new molecular entity that was for the treatment of alopecia areata. I'll give you a little bit of background on alopecia areata, tell you about the objectives of the research, tell you about the research methods. Then we'll focus a little bit on a discrete choice experiment, which is a preference elicitation experiment, which is kind of at the core of what we do, and then how we apply the results of that to clinical data to demonstrate from the patient's perspective the benefit-risk trade-offs. So alopecia areata is basically an inflammatory condition that causes people to lose hair. The largest area of hair loss, which is the largest area of hair on most humans' bodies, is on the head. So scalp hair loss is the primary focus of any treatment for alopecia areata and the primary concern for people with alopecia areata, but it can also affect hair on any other part of the body, on the face, which could be the eyebrows or the eyelashes, or anywhere else on the entire body where there is hair. So it is different from the general hair loss that people like me experience over time when your hair thins and all of those things as you age. It is different from chemotherapy-induced hair loss and those types of things. This is has a very clear pathogenesis. So the situation we found ourselves in was we were developing a treatment for alopecia areata. We'd done all our work. We're going into phase 3. The ritlecitinib, which is the product that we were developing, and baricitinib, which is the product that Lilly had developed for the same indication, are both in the JAK class, the Janus kinase inhibitors. And from the earlier experience with tofacitinib, which was also a Pfizer product, and other JAK inhibitors, there are known risks that are fairly serious, and they include serious infections, thromboembolic events, and malignancies. So the ritlecitinib, which is what we were developing, is a different type of JAK inhibitor, but we don't know yet, we don't have long enough term data yet to know whether the rates of those events are the same with ritlecitinib as they are with other JAK inhibitors. So our objective in the patient preference portion of this study was first to elicit patient preferences from a sample that looks like the indication that we're talking about. So it is adults and adolescents with greater than 50% hair loss due to alopecia areata. Once we elicit those preferences in a quantitative way, and I'll show you what those results look like, we can then use those data to estimate trade-offs that patients are willing to make. How much risk would they accept in exchange for a given benefit? We can also use those same estimates when we apply them to clinical data to say, what is the probability that the average patient would choose one over the other, in this case, ritlecitinib over placebo? And then we would look at more stochastic methods to look at the probability that these results are robust in the face of uncertainty. So we start with a discrete choice experiment. So the clinical development program is going on. We know what the endpoints are in the pivotal studies. We have identified what the risks are based on those three that are of concern to JAK inhibitors. We do the discrete choice experiment. A discrete choice experiment, I'll show you an example in a second, but since I'm speaking to an audience of statisticians, essentially it is we model the choice in each of the choice tasks that we give to people as a function of the attributes of the alternatives that they see. We refer to that often as a conditional logit model, which is a multinomial logit where choice is a function of the attributes of the alternatives as opposed to choice being a function of the attributes of the chooser. So everything here is how likely is someone to choose one or the other based on the characteristics of the one or the other. Out of that, we get two outputs. We get preference weights, which is a set of parameters on each of the, or a set of coefficients on each of the attribute levels in the model. And we can then manipulate those to get what we call attribute relative importance. And I'll show you that in a moment. I think I should probably say here that this essentially, what we are estimating is an indirect utility function. So we have this latent construct of utility that we assume is a function of the attributes of the things we choose. When we choose one thing over another, it must yield higher utility, or at least utility that is no less than the other. So we get this preference data, and we can do things with the preference data alone to understand where patients are coming from. But then we also can apply it to clinical data. And when I talk about applying it to clinical data, in this case, these are the mean outcomes from the clinical studies. We're not doing this at a patient level. We're basically saying if the patient were exposed to this profile, how would they evaluate it? And then from there, we do some calculations, maximum acceptable risk. For the benefits that this affords me, what is the maximum level of each of the risks that I would be willing to accept in order to achieve those benefits? It is the increase in utility from the benefits and the increase in disutility from the risks as they rise that exactly offsets that increase in benefits. I can use the same then. Preference shares is the probability that any individual in the sample would choose one over the other. And then the rank probabilities come from the stochastic analysis. So the discrete choice experiment. When I said I only know how to do one thing, this is the only thing I know how to do. So essentially, we take the benefits and risks in this case, or any of the features of a product or of a drug, and we break them out into their component parts, and we call those attributes. So in this example, which happens to be for osteoarthritis, we have different levels of pain and mobility, and we have different risks associated with COX-2 inhibitors. In our case, we had three benefits that were primary and key secondary endpoints in our clinical study, and then the three key risks that we have. Each one of those risks and benefits can take on a different level. So each of the benefits in this case is a response rate. So I can have essentially a low response rate, a medium response rate, and a high response rate, each with a number associated with it. And for risks, I can have different essentially probabilities of occurrence or incidence rates where I have a level associated with it. So I have this design space, which is every possible combination of levels of these six attributes that I'm talking about. I use an experimental fractional factorial experimental design to break that down into a much smaller set of alternatives that I can then show people, because the alternatives here would actually, when I think about the number of profiles and the number of possible profile pairs, I'm up into the millions. And that's just totally intractable. I can get this down to about 64 by using a fractional factorial design. So these are the attributes in our study. Hair on most your scalp, that's the primary endpoint. It's measured by something called the SALT. And if you can have a response to where the hair on your scalp that is missing is less than 20% of your scalp hair, that's considered a response. Remember, this is indicated for people who have greater than 50% of hair loss at baseline. There's also an eyebrow assessment and an eyelash assessment that were key secondary endpoints in this study. The lawyers at Pfizer have told me I need to make it very clear that although those are in the assessment report for the EU, they are not included in the label in the US. Just have to make that clear. So everybody's clear. There's no promotion whatsoever. And then you can see here, these are the levels on that far column. The levels matter in that when I do this study, I have to have a sense of what the minimum and maximum range is. So I use 0 because I have a no treatment option. And I need to make sure that the risk levels or even the response rates that I see there, the highest level that I test has to be bigger than what I see in the clinical studies. And when I don't know the answer, I have an idea, I always have to go a little bit higher so that I'm not extrapolating beyond my data when I try to apply the preference weights to the actual rates. So this is what a choice question looks like in this case. So we have the risks at the top, and then we have each of the benefits. We do a long tutorial. This is part of a larger survey. We do a long tutorial with patients to help them understand how to read these graphs and what each one of these risks entails and what we mean by each of these benefits. I mentioned before we have a no treatment option. That's often important because not everyone will accept treatment. It's much less important in something like oncology, but when you have something where there are alternatives, and the general alternatives, at least in the US, are men shave their heads and women find wigs that work. And once they do that, they do that. They're happy with that in some cases. So there are people here who are not in the market for treatment. So we have to let them opt out. If we don't, it will bias our estimates, because we're forcing them to choose something when they're not even in the market for choosing, and they can't even imagine themselves being in the market for choosing. So our sample was both in Europe and the US, and essentially, we matched in large measure what we see in the real world, which we don't always do. So we were actually pretty happy with these results. And we essentially, the only thing that was off, and this is always off in these studies, is we always get more educated people. Higher income, more education do these, almost without a doubt. Women do these more than men, and that's almost universal. So in this case with alopecia, that's not as much a problem because it does tend to impact women more than it does men. But the education level, we have to be a little bit careful about that and note that as a limitation. But that's common in most of these studies. And the AAPPO is just a measure of hair loss. So these are the results that we get from the conditional logit model that we estimate. It's actually, we have 2 different ways of estimating the model that are a little bit more intense than the conditional logit model. But regardless of how we estimate them, it's a limited dependent variable model. You can see here that the attributes are dummy coded. 0 is 0, and everything is estimated relative to that. Often you may see this as effects coded, where it's 0 centered instead. What we get here is, if 6% risk of blood clots is 0, a decrease to 2%, so that's a decrease of 4 percentage points, gives me a positive utility. And a decrease from 2 to 0.1 gives me another incremental positive utility. And the same would be true if we go from 0% hair on scalp or 0% probability of achieving hair regrowth to 10%, there's a positive. From 10 to 30, there's an incremental positive. And from 30 to 50, there's another incremental positive. Now, these are all relative to 0, which means that the increment is the difference in the size of the bars there. If I look at the biggest versus the smallest preference weight for each attribute, I get some sense of what we call relative attribute importance. Now, this is conditional on the levels that I use. If I used broader levels, I'd have a bigger relative importance. So I have to be a little careful not to go too far when I expand that. And then you can see here, and this is key to the success of this story, is that hair regrowth on the scalp, increasing the probability of hair regrowth on the scalp, is very, 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 very important to these patients. All right, so we're able first to demonstrate that the benefit matters. Nope. Did I do that? No problem. So, right here, oh, back one. There we go. All right, if I can go forward. Okay. Nope. All right, I'm actually, if this had happened to me and it hadn't happened yesterday, I would think it was because I wasn't a statistician. But since it happened yesterday, that's okay. Let me. No, doesn't. Okay. So we then take those results. Those results just came from the analysis of the survey data. So I now know the rates at which patients are willing to trade off benefits and risks. I know that on average. I can do subgroup analysis and see whether different groups trade off differently than others, but these are just the average over my sample. Then I need to have clinical data that matches what I have in the discrete choice experiment, which really means I need to design the discrete choice experiment to match the clinical data. In this case, it was relatively straightforward. Oftentimes it's not, especially if I have a patient-reported outcome instrument in there that's multi-item. It gets really messy very fast. But so here, what I was able to do from the pivotal study data was get the rates for 50 milligrams, which is what we were submitting for, 30 milligrams, which was another dose that we happened to have in our clinical program, and then placebo, and then the percentage point differences in the rates or the probabilities between those two. We used all available safety data in our submission. So I already had a source that said, okay, these are the rates we're including in our submission. I was able to have these rates then from that document and then calculate the difference in those rates for 50 milligram versus 30 and 50 versus placebo. So the first thing I do is estimate maximum acceptable risk. If I look at the rates at which we show improvement in hair regrowth, the probability of hair regrowth on the scalp, eyelash, and eyebrows, I get a utility estimate for that. Then I need to figure out how much can I increase each risk to exactly offset that utility. And I get that by this equation here. And what I'm able to show is For serious infection, risk tolerance is very high. It's smaller, obviously, when I look at the difference between 50 and 30 milligrams, because the 30 milligram dose is effective. The 50 milligram dose is more effective. So I have greater risk tolerance to make up for that additional effectiveness that I have. I have a risk tolerance for malignancy and a risk tolerance for a thromboembolic event. Now, these are 3-year risks. Sometimes, most of the time, we prefer to do these annually. This was 3 years for a very convoluted reason. Then we get preference shares. So that V that I have up there is that latent utility function that I'm estimating, right? So I have a utility function that also includes these rates for 50 milligram, 30 milligram, and placebo. So I'll get a different utility estimate for each one. When I basically plug those in, I can get the probability that any individual would choose 50 milligrams over placebo. So that's greater than 50%, and it's statistically significantly greater, and the same for 50 versus 30. So that's already telling me right now that the benefits of this product outweigh the risk regardless of dose. Then I use stochastic multi-attribute acceptability analysis to look at what's the probability that the net benefits are positive. And essentially, when I'm just incorporating the uncertainty in the clinical data, and another nice thing about this study is we had a lot of clinical data, so the uncertainty was relatively low. It's still 100% that that 50 milligrams outweighs placebo. When we bring in preference heterogeneity, which is a big source of uncertainty in our preference data, behind each one of those preference weights or marginal utility coefficients that I estimate, I assume that that is estimated or that is distributed normally across the sample, and I actually get an estimate for the standard deviation of that distribution. And when I incorporate that information in here, we see that my probability goes down to 99%. What a shame. And then when I do both uncertainty in the clinical estimates and uncertainty in the preference estimates, it goes down to 96. All of this together says that the probability that the benefits outweigh the risk, that these benefits outweigh these risks, is very, very high. Then, to compare the 30 versus 50, and we did this in response to a question from CHMP, where they said, well, 30 is effective, 50 is more effective, but why is 50 better than 30? If in fact there is a dose response relationship in the risk, which we don't know, actually. We have a lot of data on the risk for 50 milligram. We have less data on the risk of the 30 milligram. So what we did was we estimated the maximum acceptable risk is the intersection of each of these planes with the axis. So we have serious infection risk, cancer risk, and blood clot. And again, there are 3-year risks. We tested to see whether the interactions among those risks in our utility function were linear or not. It turns out in this case, very nicely, that they were. So essentially what we get is a maximum acceptable risk plane. And on that plane is every combination of those 3 risks that exactly offset the difference in benefit between 30 milligram and 50 milligram. Interestingly, Europeans are much more tolerant of risk in this case than Americans are. I would not have expected that. I'm not sure why, but as it turns out, that's the way that it is. And the red dot there is then the actual difference in risk if the risk is proportional to dose. Again, we don't have a lot of great information about the risk of the 30 milligram dose. We have a lot for 50, so we had to start at 50 and work our way down, and we almost got to 0. So essentially, we are very confident that not only do the benefits outweigh the risk of the 50 milligram when compared to placebo, but also when the 50 milligram is compared to the 30 milligram. So when we got the CHMP assessment report, I was thrilled because this is the first that I can find that I am aware of where a stated preference survey, these are not people in a trial. These are people who meet the trial criteria. These are people for whom this would be indicated, but it's a survey method. It's all hypothetical. This is the first time I've seen one mentioned in an assessment report as being important to the approval for the drug. And there were three things that we learned from CHMP's review. One, the benefits that we demonstrated. Remember that big green bar at the top when there were the green and red bars for the relative attribute importance? That big green bar demonstrated how important those benefits were to the patients. And that was acknowledged. Then the next was acknowledged. The second one is based on the preference shares. So that it is very likely that a person would choose 50 over 30, any individual, and it's consistent across individuals. And then this analysis that we did to look at the incremental benefit risk between 30 and 50 milligrams, which, remember, again, this wasn't part of our original SAP. This came in response to a question from the agency. So this was all post hoc. We had to figure out how to do it, but it actually basically demonstrated not that the 50 was superior to the 30 in the sense that they interpreted it here, but there was no reason to think that the 30 was better. So this is probably the first example or one of the first examples of how we've been able to use this preference-weighted benefit risk analysis as part of our submission to inform a regulatory decision. And it may have happened before, but this is, I think, the first time we've actually seen it in an assessment report. So we're really excited about this. And our next speaker, Francesco, and I were just talking before this. I think we had the perfect case. When we're talking about the benefit being life extension or improving quality of life for a life-limiting disease or something like that, this may not be necessary, or may not be perceived as necessary. It's necessary here because there is this sense that alopecia areata is a cosmetic condition. People learn to adapt. Why would we put them at risk for anything? And what the patients are telling us here is, I'm willing to accept that risk. And that's what was missing from the equation that we were able to offer here, apply it to the clinical data and said, even under these circumstances, patients are willing to accept that risk. Even allowing people to choose no treatment whatsoever, on average, including those people are willing to accept that risk. So I think that's why this one was successful, and we're really proud of this, and that we think this is the first of, we hope, many more. Right. Thank you.
Can you hear me? Yeah. We can take one clarifying question if there is. Yeah.
Quick question and then maybe a comment. In your discrete choice experiment for the no treatment option, as far as I've seen, you show them that there's no chance of getting 80% of the hair back over this time, but in the trial you actually have a small proportion who reach that endpoint in the placebo group. Would that create a problem then if it were otherwise? And I mean, just a comment from my side. If the result of the experiment is that patients would accept like a 5% chance of getting cancer in the next 3 years for treatment of a cosmetic disease, I mean, you can accept it, but isn't that a bit, I'm not sure, I have difficulties to believe then that patients understand the risk actually.
So there are a couple of questions in there. I'll start with the last one. Patients understanding risk is something we have to spend a lot of time on. So remember, this is a survey and we introduce each of the attributes in what we call an information treatment. And we have to make sure that they understand the information that we are presenting. Now, we're limited in how far we can go. We can't give them a whole university course on it. So we have to do it in a short amount of time, but then we also include comprehension questions and test for whether those who did not understand versus those who did, who got the comprehension question right or wrong, impacted the results at all. So we do test for things like that. In terms of the opt-out, the reason the opt-out is at 0, and this is a fairly strong assumption. In the end, I don't care about the opt-out being 0. What I care about is what is the preference for a difference between 0 and the next level. And what is the trade-off between 0 or between the second level and the third level, right? So that anchors me to a place to start. And I'm really looking at the differences. So when I then apply this to the clinical data actually have the placebo and the 50 milligrams. So I have a non-zero level for those data. What I have to assume is a relationship between 0 and the next level, and assume essentially that that is linear. I can test that. And in this case, we found out that it was, which gives us some confidence that we don't have a problem with setting that 0. Sometimes we do. Sometimes 0 really does matter. In this case, it didn't seem to.
Thank you.
Thank you. Simon Wandel, Novartis. This is really fascinating, and I think maybe there are two aspects you could comment on. One, you elaborated a bit, right? So the patients who take part in your preference study may not be the same that would be in your clinical trial, right?
Right.
And so you can wonder, is it really, how do you bring these two worlds together, right? Because they may really be different populations. And to that point, you also went into this, could you comment a bit more, like men are less likely to take part in your survey, right?
Mm-hmm.
So did you address that with something like inverse weighting, or did you do oversampling, or how did you go there?
So in this particular case, we got lucky. And that is because the proportion who participated in the sample were roughly equivalent to what we see in the real world for people with alopecia areata. Right? So in that sense, we kind of matched up. Normally, we don't. We don't necessarily weight these, but we will test. So we will see, is there any significant difference between men and women in their preferences in our data? If there are, we need to make an adjustment for having that imbalanced sample. If not, we assume we're okay. And that's an assumption again, but we use that as the basis for that assumption. As far as the patients go, we try to match the indication as much as we can, even more than the clinical trial population, right? Because it's the indication that we're trying to think out toward, the people who are actually going to be exposed to a drug. And we have to get that right or get close to that. Otherwise, we need to reweight. We do assume that the preferences are consistent between those in a trial and those not in a trial. Now, ideally, I'd love to do these in clinical studies, but ClinOps doesn't like that very much because it costs money and disrupts their trial, and they don't like that at all. There are some cases. I happen to know a colleague at Johnson & Johnson is doing things in trial probably more than anybody else. There may be others, but I just happen to know that he's doing that. It would be nice to see whether in trial and out of trial is different. That would be a great experiment. We just have to convince the clinical teams to allow us to do that. And that's the biggest challenge, frankly. So.
Yeah, I think we should stop.
Okay.
Yeah. So thank you again.
Yeah, thank you.
So very interesting to see a real world case where patient preference actually gets on the label. We'll now move on to ICH E22 and general considerations for patient preference studies. And it will be by Francesco Pignati. He's a medical doctor, a Master of Science in Biostatistics, and he held roles at the European Organization for Research and Treatment of cancer ERTC data center until 1999. Since then, he has been with the European Medicines Agency, EMA, currently serving as scientific advisor for oncology and focusing on cancer drug regulation and benefit risk analysis. So the floor is yours.
And this would all result in endless discussions and difficulties. And so what we've done over the last 20 years or so, we've tried to make this benefit risk assessment more systematic. And the E22, which is this new guideline which is being developed at the level of ICH on general considerations for patient preference studies, is actually the result, the sequel, if you want, of the implementation of the benefit risk assessment. And really the trick has been for people to understand that when they make a statement like the drug is better than placebo, they're actually making a preference statement, but implicitly, they're just saying, I prefer drug to placebo. And this is not terribly transparent. And what we've done is to push our assessors, to push the companies to build their argument in a structured way, and not just say holistically, benefit risk is positive, but attribute by attribute, benefit and risk by benefit and risk, say why the drug is better than placebo. And basically, it means going from this statement here to something like an explicit statement that you see down there, where you need to break it down at the level of the benefits and the risks and assign some weights to these quantities. And this has taken a while because it's much easier to do it implicitly and intuitively, and then to have to justify it and so on. And many of our assessment teams, and for sure, in the case of what you've seen, assessment teams really find it extremely useful to discuss weights and preferences in a model more than these holistic discussions, which are not particularly scientific or constructive. So structuring the benefit risk problem, identifying that there are weights, subjective weights in there, value judgments, that was the first Herculean effort that we did. And the second was then, hang on a minute, subjective weights, what is this? We are doing science here. This is going back to the dark ages where, you know, we were talking about hopes, about, you know, what we wish from a drug. And the whole point of drug regulation was that patients and doctors are unable to assess the drugs. Now, why on earth would you go back to that? And really, with some friends, we call this the Pandora Project. It's like opening up the Pandora's box of asking patients, well, what do you think of these effects and so on? And so the second effort has been to try to explain why it is important to actually discuss patient preferences. And they would accept that their own preferences could be discussed, their own value judgments could be discussed in some such frameworks. But why patient preferences? And the most conservative would call them biased or unduly influenced by perhaps by promotional information and so on. And so to convince them of this, we need to, and this is an important point that you may have to work on in your organizations, in industry, to convince people why it is important to have the patient's view on this. And I'm just focusing here on the regulatory decisions. Forgive me just for brevity for this talk, but there are many other uses that one can do. But for regulatory decision, for regulators, why on earth do we need the patient preferences for the regulator? And this depends then what type of regulatory decision you have. And let's just focus for decisions around approval and decisions about product information. So I'm just going to focus on there's many more where patient preferences and other preferences could come into play. But the first of the thing that you need to decide is, you need to know, you need to ask your regulators, is this a situation where you're likely to find an attitude of regulator knows best? The values of patients, well, they are what they are, but actually, you know what, I know better. Or a situation where the regulator describes the effect, we scrutinize the science that produces data, but then with good information, we're basically going to leave it up to the patients to decide. And depending on the situation, regulators take different, let me call them philosophical approaches to these type of questions. And different regions might be different. So you need to know what kind of regulatory decision, what kind of regulatory context you're looking at. Now, regardless of this context, there is more or less use of patient preferences. Even in a directive situations, I know best what patients should want, the patient preference will provide the context for the regulator's preferences. So as a company, you might be able to argue for or against a certain attitude by the regulators. Regulators themselves might be able to say why their views are different from those of patients and justify any difference, and so on. And in situations of more or less, let's say, equal situation, then the preference studies may tip the balance in one direction or the other. And so in any case, it will increase public trust not just in the regulatory decision, but also in the work you do as a company. When you come with your dossier, there you go, benefit risk is positive. No, you will have science to support your statements. In situation where this is more of an informed choice, Brett was mentioning situations where there is almost infinite acceptance of the risk, like certain desperate cancer situation. Well, there the usefulness is more theoretical in the sense that you let them decide, so it doesn't really matter for the patient to know what the majority thinks. It's still an individual choice with their doctor in the end. So then it may be less critical, but still informative for the regulators if the utility is not present. What type of drug are we putting on the market here? Is it the whole majority of patients who think this is a wonderful drug, or actually it's only a small niche? And in that case, and that brings me to the third column of this table, it's very critical information for the product information. In fact, all of these can be used to support writing risk management plan, communication strategies. If there are groups in the population who have extreme preferences, you were mentioning the situation of somebody who would accept any risk of cancer for treating alopecia. Well, then you know how to target a certain population with the proper information they need for making a good decision. So actually, if you think of it, regardless of the role in the benefit-risk decision, patient preferences have a clear role in guiding proper risk management and proper product information, okay? So this is a lot of the discussion that has brought us to finally engage into a new ICH guideline. It's not a simple task. The last thing that we had to look at was, well, what is the methodology for these patient preference studies? They come from fields like behavioral psychology and economics. They're not speaking the same language as our, let's say, pharmaceutical statisticians. So one of the things we will have to do is, first of all, develop the guidance for what are more or less observational studies. This is still relatively underdeveloped within the ICH and within also our own guidelines. And also develop a language which is close to the E9, to the Estimans language, and that will fit well, let's say, within your current thinking and structure. Now, there will be a learning curve for all of these methods, and this is something that you will have to go through. Now, luckily, a lot of work has been done by many of you as well. In Europe, we had recently completing the PREFER project, came up with recommendations and tons of information on how to do these studies and how to look at the different methodologies. This doesn't come in isolation. There's many more produced for drugs, for devices, by regulators, ISPOR, and others. So there is a huge amount of material, and now the E22 guideline is likely to pick the high-level principles from all of this material and turn them into an ICH guideline. We have an expert group of eminent scientists. They're currently working on building a consensus, drafting the first technical document. The public consultation should be before the end of next year. So you will see something coming out of that. Again, even the title tells you this is about general principles, and so you shouldn't expect anything incredibly revolutionary, and most of it will already be in the guidelines that you've seen. There is already a concept paper which tries to delimit the scope a little bit. Besides being high level, it will draw extensively from the established framework. We're looking at evidence that will add, not replace, the existing clinical evidence. It will not likely be mandatory, but it will be highly recommended in many situations. It will not only be talking about the benefit risk assessment and the regulatory decision, but also the huge importance of looking at patient preference in a systematic way to inform the drug development. Obviously, drug development going hand in hand with the purpose and decisions. What also some of the regulatory authority were adamant to stress at this stage was that we should not address labeling issues, how you get this data into the label, how you're going to make claims. That's kind of regional. Internal kitchen that ICH should not touch, and good with that. All right, so in summary, new guidelines, you should expect to see draft documents in a year or so, but until then, there is a time to familiarize yourself with the vast material that is already available from the authoritative sources like PREFER, FDA, ISPOR, and the devices. There will be challenges for you, for your colleagues, familiarity with the concept, applications. We've seen one example. We would like to see many, many more of this kind. And like any other field of medicine, science and methods which are evolving, for us, the challenge of how to keep it simple with so many angles available and how to bring all of these methodologies to the language of the that we are used to. Thank you very much.
Thank you very much, Francesco, also because you are in time, perfect in time. We have time for some question or clarification question.
Thanks for the presentation, Francesco. So you mentioned that for the ICH guidance, the preference studies and labeling is going to be out of scope because that's regional. Would you, however, like to comment on the EMA thinking of preference data in labels?
I can't really think about the EMA thinking because we haven't published anything on that. But my own thinking is that initially I found it very difficult to imagine how a patient would care about how the population thinks about a drug, because the decision will be very individual. So from that perspective, some sort of star rating, like when you're purchasing something and know how many have reviewed this drug and think it's a good thing. I don't think that's the way to go, but the way to go is the discussion that you have with the doctor. And then I think there is a totally underexplored role of regulatory information in the doctor-patient decisions. It's officially outside our remit, and there are other organizations and governance which look at the way decisions are made at the level of the patient. But I think the regulator should provide information, regardless of the model, shared decision-making or whatever, for them to make then the right decision. And then we should focus on producing that information. So perhaps also in the label, if there is I don't know, a group with an unreasonably high risk perception that or groups that know or when you know what are the critical questions, the critical attributes that people will look at in order to choose whether the drug is useful or not, this would be very useful information to communicate even in the label. So from a perspective of decision aids, I think there is certainly an opportunity to use the label for that.
Okay, thank you. I want to introduce the next speakers. We have a joint talk taken by Elina Azikanius and Muna Akacha. Correct? Okay, good. Elina is a statistical assessor at Finnish Medicines Agency. She joined Fimea in 2020, and before that, she worked over 12 years in the pharmaceutical industry. She is a member of the Methodology Working Party and Scientific Advice Working Party, and a member of the Biostatistics Interest Area Leadership Team. Just also, I want to introduce Muna Kacha, is the global head of the statistical methodology group of Novartis Pharma, based in Basel. Muna has a wide range of research interests, including topics on estimates, missing data, longitudinal data, recurrent event data, and those finding studies. Before joining Novartis, Muna studied mathematics at the University of Oldenburg in Germany and was a PhD in statistics from the University of Warwick in UK.
Thank you.
Thank you.
Yeah, so I think Sheila's question was the perfect segue to this discussion. So we are now talking about, in a way, communication with Muna. How do we get the information that is relevant for the patient out there into the kind of public domain, right, out of the clinical trials? But not to be vague and high level, we have chosen now the label, the SMPC 5.1 as our kind of focus point, and what does good look like. This is my disclaimer, so my personal views. So what is the SMPC? I think most of you know and have some hands-on experience working with it, but maybe not everybody. So it's a document describing the properties and the officially approved conditions of use of a medicine. Summaries of product characteristics from the basis of information for healthcare professionals and how to use the medicine safely and effectively. This, of course, does not mention patients. It mentions the healthcare professionals, but in a way, it should then somehow form the basis for that conversation that that healthcare professional has with the patient. There is 3 sections that I have included here that we as statistical assessors often are involved in thinking about. First one, in a way, and the most important one in a way, is the section 4.1, which includes the indication. So it includes for whom is the patient, the drug is actually indicated for. It includes the population, right? Or if you think on more complex ways, like yesterday, the discussion about adjuvant and neoadjuvant treatment and so on, is it for all of it, adjuvant only, neoadjuvant only? So it really includes that thinking as well. There is the section 4.8, which is very important, but not the focus today. It includes the undesirable effects. So basically the side effects, the adverse events. And maybe we actually should put more focus and emphasis on that one as well. And then there is the section 5.1, which is called pharmacodynamic properties, which in plain English means the efficacy data for us. And I have here this kind of 2 dimensions to this thinking, whether the data is relevant and whether it's reliable. And there is, of course, a lot of different words one can use, especially for reliable. Is it statistically robust? Is it maybe not biased? Is it, but what I mean here with reliable is that can we trust the results? Are they trustworthy? Can that healthcare professional inform those treatment decisions for the patient, discussed together with the patient based on that data? Is it reliable enough? As we are now discussing here, the 5.1, that discussion is not relevant unless there is not a positive benefit risk, right? So we are not discussing in a way that part because there is a label only if the drug is approved, right? So now we go from the assumption that benefit risk is positive, however it's defined and assessed. And I think that in a way, the statistical assessor job is to ensure that the information in 5.1 is reliable. So that's what we do when we work as a statistical assessors at the agencies. We are not necessarily experts on what is defining on what is relevant, though we may have some opinions, right? And of course, some of us are patients and, we have all sorts of background, right? But that's not in a way our job. But our job is to ensure that the information in 5.1 is reliable, that you can trust it. And unfortunately, often we see that the relevant endpoints, information that is important for the patient, has been collected or analyzed in an unreliable way. And hence, It will not be included in 5.1. And this is basically my plea here to the industry statisticians, that make sure that the data that is relevant and important for the patients is also reliable, because we don't want to exclude data from 5.1. We want to include all the important data there, right? We also think it's important. That's not the point. So what do we look at? And this is a list of things. Things are not black and white, and this is for sure not a list of tick boxes, right? That everything has to be fulfilled and completed in a perfect way, and only in that case data can be included. This is things that we look at. And of course, we always start with the study design. Is the study designed to answer the question of interest, right? Is the study actually suitable for basically analyzing that endpoint data point that we now want to include? And are the data collection methods and the analysis methods aligned with the estimand? So with that question of interest, is the study design suitable with that? Is the schedule of assessment suitable with that? And are the statistical methods suitable with that at the planning stage? We look for type and error control, multiplicity control. Surely your chances are increased if multiplicity is covered. We look at things that make the study results reduce bias, randomization, blinding, objective endpoints, the basic things, right? But we also understand and acknowledge, for example, when it comes to objective endpoints, that also subjective endpoints can be important for the patients, right? But then the bar is higher, right? Then you need to do more to get those subjective endpoints into the 5.1. Then you need to kind of get more on the green side on the other aspects, right? Because of course, not all patient-relevant endpoints are objective. We want to see pre-planned sensitivity and supplementary analysis. So things that you have pre-planned, what could happen and how do we fully understand. Yesterday, there was a lot of discussion about estimates, right? How can we in a pre-planned way, anticipate different analysis than the primary one? We look at descriptive statistics and visualizations. So even though a lot of effort has been made in defining the primary estimate, that's not the only data piece that we are interested in. We want to see how those things came together. Because something that I feel now when we have put so much emphasis on the estimate thing is that we are not putting enough emphasis anymore on the basic stuff, right? Some, graphical displays of the data, us understanding how patients move from one category to other, things like that. It's really simple things. We hope for adherence to guidelines and scientific advice if scientific advice has been sought for the program. But again, you can also do something else that has been defined in the guideline, but then you need even a better rationale, right? And then if you use the endpoint from the guideline. Okay, now we switch from planning to things that happen during the study. Minimization of bias. And good study conduct is my favorite one. So now you need to follow the protocol. You planned the perfect protocol, but now the statisticians need to sit with the team when the study is conducted and tell, do not let these patients drop out from the trial. Don't let them drop out from the trial, right? So really explain that how those things that are defined in the protocol are actually important so that you get those reliable results out of the study. And it's not only dropout, but that's the best example. And then when we look at the results, of course, we want to see convincing results. We want to see a big delta, because then if we see a really strong treatment effect, then all the uncertainties matter less, right? If the treatment effect is really strong, then all these other things in a way matter less. We can trust it. We look at imbalances in patient disposition and in the current events, and I'm really, really surprised that these are not included by standard in most of the CSRs that I look at. So again, it's not that you define your primary estimate and you define your intercurrent events doesn't then suddenly mean that we're not interested in how the distributions look like, right? We also too often see obvious missing tables and incomplete information. And I think this is especially for stuff that, again, happened during the trial, so that we would exactly try to now understand, why did those patients drop off? Why is there so much censoring on the PFS curve? Why is there so much censoring on the OS curve? What is happening, right? So these are for me what I mean with obvious missing table is, for example, a good example is of reasons of censoring not explained. Are the patients censored at cutoff, or did they drop off? And if they dropped off, why? Toxicity, withdrawal of consent, what was the reason? And we also look for interpretation aligned with results, because that brings me back to the most important point, which is transparency. We want to read the representation of the results the way that we have the feeling that you are not hiding anything from us, because we are looking for trustworthy results, and the results need to be reliable. So transparency is key. So then I have here a few examples. They are made up examples because for obvious reasons, I did prefer not to use real cases. So the first one is about a composite estimate. And what I want to highlight here is the point that in the current events carry patient-relevant information. So these are the key results. You have some sort of responder endpoint at week 52, 20% of responders in placebo, inactive 50%. It looks good, right? There is 30% difference there. 30 percentage points. But then if a naive person, I guess I would think, looks at this, and you have the proportion of non-responders, you would then think that that means that those patients did not respond. So they didn't have the response. But actually, when you look at what does that consist of, you get all sorts of stuff, because it's a composite estimate. And all the stuff that happened is put into that non-responders category. So you have clinical non-responders, so the true non-responders, you have dropout, you have rescue medication use, and you have AEs leading to withdrawal, right? AEs leading to withdrawal, you only have in the active arm. It may be important for the patient to be really tell that actually 10% of the patients did not reach this endpoint at 52 because the drug was too toxic. Or it may be important to tell that the rescue medication use was actually, and usually those are toxic, right? There may be things like corticosteroids, was quite balanced between the arms. And what is that random 30% dropout? But to be honest, I don't know how the patient could understand that either, so it's better not to have it. So good study conduct is key. But how the same table could come another way is that you actually see that 60% of patients in the placebo group used rescue medication, which could be something really toxic, right? With really big side effects. So the second table is actually, I think, strengthening the treatment effect. The treatment effect of the active drug is actually even better than those 30% that kind of tell me, because so many patients are on this toxic rescue medication. So this in which box we are with this drug is important information to be included in the label, in my opinion. So then I have another very common example here. We have a time to event endpoint and early dropout. So the first one is kind of easy. 10% of patients withdrew in the active arm through to treatment-related infusion reactions, so tolerability issues. Important information. And maybe the treating physicians can even do something for it, right? With planning careful patient care, maybe something can even be done in practice that the patients can continue the treatment. Other easy communication and important communication, 10% of patients in the active arm, the CAR-T therapy was not successfully manufactured. So the actual treatment was not produced for them. Again, important information. The last one, personally, I find difficult. 10% of patients in control arm withdrew from the study immediately after randomization. So, and then what? What does that mean? It doesn't mean anything. Nobody can understand that. So again, things like that should really be, in a way, prevented, because even for me, whose job is to assess that kind of information, that information is really difficult, right, to do anything with, to understand how does that actually impact the results. If you have approved the drug, because we are here in the positive benefit risk setting, we have clearly taken into account that we can trust these results enough to approve it, right? But still, from communication point of view, the difficulty remains, right? That's not a way. So looking for good proposals there and hope to see them in the dossiers. Something that I also see is a bit problematic, that we have the tendency to kind of oversimplify complicated things. So this is just, I took this just from Google, this graph. So it's on cancer drugs on PD-L1, right? Where we know that PD-L1 is a predictive factor, right? It's not, if you give the patients a PD-L1 inhibitor, then the PD-L1 level, it does impact the outcome. And then we still see often sentence which say that similar efficacy was observed in all relevant subgroups. And I'm like, no, it was not. But the benefit risk can still be positive. So again, if you're approving the drug for the whole indication, irrespective of PD-L1 status, we think that the benefit risk is still positive for everybody. But still, it can be important information to say that, you know, if your PD-L1 status is above this cutoff or whatever, you have a better prognosis than if it's below that, because patients probably really care about how long they will live. So it's probably important for them, though the benefit risk is still positive across all the levels, right? I mean, my text here is not perfect. It's just an example to trigger the thinking that sometimes we want to see things in an oversimplistic way. And what I meant is with this kind of trustworthy communication, that then if we as regulators see something like that similar efficacy was observed in all relevant subgroups, and it's so evident that that was not the case, then we start to question every sentence that you have written, right? So don't oversimplify things. Okay, so this one is a little bit a complex one. But I wanted to have an example where the study design really has an impact on whether the data is included in the SmPC or not. So this is a placebo-controlled trial for a chronic disease in an approved indication. So it's in an approved indication. So there is already, the indication is already there. Primary endpoint is response rate at week 52. It's a composite estimate. And the protocol includes a very strange sentence that in both arms, placebo included, patients may move to double dose of the active treatment after 22 weeks, which was kind of the induction therapy, in the absence of benefit. It's very strange, but the drug is approved, so they know it works. So it's probably some kind of motivation for the patients to join the study. But now you're totally destroying everything, because you let everybody to move to something else than what you wanted to study before you're assessing the primary endpoint. Okay. 40% of, because maybe this wouldn't matter if nobody used this option, right? But 40% of patients in the placebo group switched to this double dose. So it has a large impact on the results. So what do we do now? We don't, so maybe we don't want to include the results in the SmPC at all, because we don't think they're trustworthy. And if we included them, we would only include them if we can discuss this point about the double dose. But because the study was also not properly designed to study the efficacy of this double dose, they wanted to have the double dose as an approved, like if there isn't enough response, you can higher the dose, but we didn't have enough evidence for that. So we can't include that in the posology section of the label. We also cannot describe the double dose in section 5.1 because that would be off-label use, and that's not okay, right? It's a bit complex, but like the point is that if your study design is not optimal, it could maybe still be okay if the things didn't happen in a data-driven manner. But when then they happen, we basically decided to exclude this whole RCT from the 5.1 because we could not find a way how to in a reliable way communicate about the results. So my take-home messages are that the statistical assessor is the filter to ensure that only methodologically reliable information is included in the SmPC. Whether something is reliable or not is not black and white. Unreliable endpoints may be included in the EPAR where there is more space to discuss things. So EPAR is this European Public Assessment Report that has been mentioned many times. And the burden of the proof is on the side of the industry. So we know, including 5.1, what we know is reliable and relevant, and not what we cannot be sure of being unreliable or irrelevant. And now I move to Muna.
Thank you very much.
I'm accepting the challenge of having slides in 2 minutes, but I'll try to, yeah. So hi everyone, thanks. And thank you, Lina, for setting the stage so nicely. So I would like to follow up with a couple of thoughts on reliable
and then also say a couple of words about SMPC. So we saw several examples about reliability. Is it reliable or not? And as Elina has said, if we ask this audience, what do you mean by reliable? We will have several different definitions, I'm sure. I think generally for the discussion, it's quite helpful to carefully distinguish between, do we talk about reliable endpoints, reliable data or analysis? And within these slides, I want to focus a bit on reliable analyses. And I think although we all have different definitions for what we mean by reliable, I think often implicitly or explicitly, we compare in our minds to the gold standard of evidence generation, RCTs. And just as a reminder to all of us, why is it the gold standard? Well, it enables us to estimate the causal effect of treatment assignment, and maybe more specifically or more precisely, actually, that only holds in the absence of missing data as well as mild assumptions. And the superpowers, let's say, of RCTs come from the randomization, so like with right comparison, an internal concurrent control, but also that we have a tightly controlled experiment. Now, the examples by Elina have shown there's nothing like a perfect RCT, right? And I'm sure all of you working on RCTs have never seen a perfect RCT where, for example, no patient dropped out. So what are the considerations as we move away from a perfect RCT? And I personally found this paper quite helpful by Hernan in 2013, where he tries to essentially give this a bit of a comparison to observational data. They say, the authors, what started as a randomized trial effectively became an observational study that now requires analysis that's complement and go beyond intention to treat analysis. And then he goes on by saying the key obstacle in the adoption of these methods is the widespread reluctance to accept that overcoming these limitations necessitates untestable assumptions. I'm sure all of us can relate. And they also go on and say that embracing these more sophisticated analyses will require a new framework for both the design as well as the conduct of randomized trials. So I draw your attention to the date of the publication. It was in October 2013. Yesterday, we heard that the concept paper for estimates and sensitivity analysis was published sort of 1 year later. So one could argue, well, maybe estimates and the framework as expressed in the ICH E9 is actually this framework that will help us to actually have these conversations. And I think indeed it does, right? It does provide us with a nuanced language to express scientific questions of interest more clearly. And it also provides us a framework that stresses the role of sensitivity analysis. So using the terms that Elina has laid out, you could say it provides us a framework to really discuss relevance as well as reliability. And we have seen that the discussions like yesterday, so we are having the discussions, but I think it's also fair to say that we as a community have a bit of a challenge, let's say, to try and consolidate these things and to discuss these on equal footing. So Elina has said we are not experts in defining what is relevant. I would fully agree. I don't have a medical degree, and even if I had one, it would be probably for a certain disease area, and you can't be an expert on everything. But I think we also have to be very mindful that the thing that we know about, namely how to design trials, how to analyze data, and so on, impact relevance quite heavily. And we all have seen this graph from the training material on the estimate framework, right? So by taking certain choices, we are impacting the relevance, and what is relevant may not be reliable and vice versa. And this always brings me to this quote by John Tukey, who says, far better an approximate answer to the right question, which is often vague, than an exact answer to the wrong question, which can always be made precise. So what are the right questions when we think about the SMPC and the label? And we have already heard some nice presentations today. We also discussed it a bit yesterday. And of course, it will very much depend on the therapeutic area again. Anna will talk to us also a bit about what relevant questions, let's say, from her perspective is. But I think it's important for us to have the consumer in mind at the end. So it is for the medical doctors, let's say, ultimately for the patients. So what are the right questions for them, and how can we answer these questions? But I think there are additional questions for us as a community to answer, because to be honest, to me at least, it's not even quite clear how I get from an approval and the evidence and the information that is provided for the approval, so the thumbs up, how I get from that to a user manual, so to the SMPC4. What are the rules that get me from one to the other? Certainly there are some roadblocks, let's say, or some good reasons for not including certain things, but it's not always very clear if these are set out that clearly. And I think we have already discussed some things. For example, if we have a composite endpoint, what do you put in the label, right? Or also if you have a treatment policy approach for your estimate, what do you communicate in the label? The whole policy? Is it a label for your drug, or for the policy, including rescue medications and so on? So thinking about reliable as well as SMPC, my last slide is, I think clearly I'm also part of the community, right? So there's also tension when I think about these things. Like on the one hand, I'm a statistician. I like rigor. I like making as little assumptions as possible. All of us fear and hate untestable assumptions. I get it. But we are also, I think, patients and caregivers to a certain extent. And if we go to a doctor, let's say, do we get all the answers that we hope for? Personally, I think we can do better, but it's not easy. So if you, for example, went to a doctor's office with your child or yourself, you're sick, and you ask the doctor a relevant question to you, and the doctor says, well, I don't know because the label doesn't give me this information. How happy are we with that, as opposed to actually the label tells me something about it, the answer is only vague or approximate, to use the terms that you can use, but at least I can give you some information, right? And I think we have to discuss that. In terms of a potential part of a solution, I think what could help us as a community is to distinguish more clearly between testing and what do we want for testing and what is reliable testing versus estimation. Also, I think we should be happy to say something is reliable when it provides, when I assess it through sensitivity analysis, let's say, across a range of plausible assumptions, and the conclusions hold strong. So I don't think we would be well advised to say something is only reliable if it doesn't require untestable assumptions, because then there's very little we can do, right? But I think for that, it's also important for us to do maybe a better job at pre-specifying what we mean by plausible assumptions, because then we can go back to that and we don't have to discuss worst case imputations and things like that. And finally, I really think we can learn a lot from the causal inference community, who is very used to deal with very messy data. The good thing is, so the causal inference community doesn't necessarily have the luxury to design trials the way they want. So now if we combine our powers that we actually design trials and experiments with the superpowers of the causal inference community, I think bringing these two together can actually help us to provide more relevant information to patients and prescribers. It wasn't 2 minutes, but I tried hard. Sorry.
Thank you.
Thank you. So.
So in the interest of time, I think we'll save the questions for the panel discussion afterwards. So we'll just go directly to the next one, which is now finally a patient representative. Anna Wiksten, she's a PhD statistician, has worked the past 15 years in various drug development projects as a statistician and statistical programmer. She has a broad experience in indirect treatment comparisons, regulatory submissions, and estimates. Beyond her role as a statistician, she is a patient and patient advocate with the desire to understand how we can improve drug development process from different perspectives. So,
thank
please.
you. Does it work? I guess. Yeah, so first, thanks for inviting. It's a great honor to be speaking here. And speaking about these two perspectives. So let's start. My disclaimer. So all views are my own, and they don't present any of my employers, any patient advocacy groups, and also not necessarily any other patient. And I'm presenting things as I perceive them as a patient using my statistical knowledge. And I made mistakes in both roles, and I made also learnings in both roles. And I hope with that I can share you something insightful. Here is my acknowledgement. So So there were some really great people helping me with this presentation, so thank you. And then, yeah, here is just a little bit about my background, but that was already in the introduction. So let's start. I will use in this presentation thought bubbles as my thoughts as a patient. So how I remember, I thought about different things when I experienced them and so on. And the content of my presentation is patient journey and prognosis. Then I will speak a little bit about being a patient at work. And then about relevant data where I reflect certain topics on my patient and statistician perspectives, and conclude with summary. So let's start with the beginning of the patient journey. So 2017, in September, I had a stroke. It was not bad. I was 1 week here in the Basel University Hospital. Then I went back home, and after 2 weeks, I was back to work. A month later, they found the cause for the stroke, and that was cancer diagnosis. And 2 months later, I started then the treatment. And that was a difficult choice, I have to say. That was from those plots, that was the most difficult moment to think about what do I want to take. And my doctor introduced me two options, and he didn't tell too much about the treatments, but I did a lot of Googling. And in my mind, I found that treatment A is 50% risk of depression and some other awful side effects. Treatment B, 5 risk of leukemia in 20 years. And this option my doctor didn't give me, but I kind of like, I of course have always the option to take no treatment. And then I might have an elevated risk of a stroke. Yeah, so not an easy choice. My initial preferred order was this: no treatment, treatment B. So I was like, rather something really bad in 20 years than something really bad now. And treatment A, it was kind of the depression risk was really the one that was worrying me, and why I was so hesitant to take it. However, my treating physicians and the medical team's recommendation was treatment A, and I decided to go with that, try it. And here a little bit about the disease and treatment. So I have a blood cancer, which causes my bone marrow to produce too many platelets, which then cause the stroke. And then when you have had a stroke or thrombosis, you are a high-risk patient. And then the recommendation is to start treatment A or treatment B. And treatment A is affecting the platelets. So my disease is my official diagnosis is prefibrotic myelofibrosis, which manifested for me as elevated platelets. And it can progress to myelofibrosis. And myelofibrosis is fibrosis of bone marrow. And at late stages, the bone marrow stops producing blood cells. And we all know what happens if you don't have blood cells. So really not a nice disease. And again, I started to do Googling of Kaplan-Meier curves. And I found that median time to progression to myelofibrosis would be 3.5 years. 3.5 years, and then I found another Kaplan-Meier curve, which was median time to death in myelofibrosis, 3.5 years. So that was 7 years what I counted. And yeah, my patient perspective, this prognosis is really poor, and I might die at the age of 40. And yeah, this hit and triggered quite a thinking process. I mean, first, I was simply sad. I mean, this was not what I expected from life. But then I started to accept it, and I was like, how do I want to use those 7 years? 7 years, and then I didn't have too much of in my mind that my bucket list was kind of empty. and I started to think that actually I have already lived a full and happy life. So I keep doing the things which made my life full and happy. So I accepted the situation. Then poor prognosis, my statistician perspective. Well, obviously it didn't hold. So the 7 years have passed, and I think I might be alive. Then it didn't take into account all available medical evidence. It didn't take into account my baseline characteristic. It didn't take into account current and future treatments. And as a statistician, I should at least the 2 first bullets I should kind of consider when I do some prognosis or prediction. Then in 2018, I did a better prognosis using real-world evidence and a sign-up. So I found this paper published in New England Journal of Medicine, classification and personalized prognosis in myeloproliferative neoplasms. And I, of course, then filled all my data in and checked what does it give to me. And suddenly my expected median event-free survival was more than 25 years. So quite a bit more than 7. And my patient perspective was, I mean, that I might live long after all, and that is amazing. And I kind of I compare this feeling that when I am having a flu or something like that, then when I recover and just feeling normal feels kind of energized and happy and so on. And this is the same feeling, but then it's like exponential, and it also seems to be like lasting. So that was amazing to get that prognosis out of that Shiny app. Then my statistician perspective. This is the coolest Shiny app I've ever seen. And they published it in New England Journal of Medicine. So that would be really cool to have a Shiny app in there. But then I started to ask questions. How did they implement it? What kind of models they used? And what happens if I change the input? So changing the input, I played quite along with the app, but then one thing was really interesting. So looking at prior thrombosis yes or prior thrombosis no, and otherwise keeping everything the same, was that why is the prognosis better with prior thrombosis? So why does the stroke improve my prognosis? And I do acknowledge the difference is minimal, and there can be many biases, and it can be by chance, but anyway. And this, of course, then triggered a question in my statistician mind that you remember the treatment A is given to high-risk patients who have had a prior thrombosis. And this triggered a question in my mind that could it be that the treatment A is also affecting the cancer? And I don't know this, so it's remains unknown, but kind of. And then combining the perspectives, what I kind of learned from doing this prognosis to myself is that the data can be really powerful on its own. So when I did the poor prognosis, it led to acceptance. My time is short. It is okay. And how do I want to use it? Whereas then when I did the better prognosis, it led to hope. my time might be longer, and it is awesome. And how do I want to use it? In a way, actually, both of them led to how do I want to use it, and then some kind of peace of mind, which is also valuable. And obviously, these prognosis didn't change anything in my treatment, but they still impacted a lot me as a patient. And I discussed this with one of my doctors and he said it very wisely, that kind of, if you can't treat the patient, you can at least tell the truth. And we as a statistician can make impact by data. And this really changed my thinking that I mean, I am a drug developer, when I work in pharma, but actually also purely the statistics can be really impactful if it's relevant and so on. Okay, then I will speak a little bit about being a patient at work. So I was changing job same time. So the week before, it was Monday morning when I had a stroke, and the week before last week I had a job interview. I got the job. Then I went through all of this, and beginning of December, I was really tired, and I remember I was going cross-country skiing. I mean, so I was physically doing very well, but all of this was still emotionally tough. And I was really tired. I was sitting on a bus stop and just started to cry that I want to have another stroke so that I can go to hospital and rest. And then I kind of understand that now I'm not well and it's time to go on sick leave. And I also decided that I will inform my new employer the same day as my sick leave started. And this response from my new employer turned out to be one of the biggest turning points in autumn 2017. And why was it so impactful? It came. So sometimes it happens that you get no response. And at this I have heard from other patients as well. Then it came fast. So it came the same day. I didn't have to spend 1 week waiting that if this manager has time to respond to my email. It was emphatic. It offered support. It asked, what do I need? What do I or does my doctor think? So the person didn't think that she has the answers. And it encouraged me to contact again if I need anything. And this was also really, I mean, obviously it was a big decision for me that do I tell this or not? So the person kind of understood that and gave me way to be open in my communication. And then what is also important, there were things which it didn't include: pity, assumption, or unnecessary help. And these are, I mean, pity is something which is very difficult for me because, I mean, of course, these things have been difficult, but I don't want others to make my life look worse than it is, or kind of comfort others that, hey, I'm just okay, or so on. And also assumptions are difficult because we never know how things impact others, and these are complicated things. And then the last one, unnecessary help. This happens actually quite often. And of course, it's well meant, but it also takes your energy if you need to kind of be rejecting some help which you don't need, and telling that I don't need this because blah, blah. And my advice is for patients to inform in a written way so that the other patient have time to digest. So it's probably also not easy for a manager or a colleague to receive this kind of information. And then for leaders and colleagues, respond timely with empathy and asking what is needed. And of course, I had the big question in my mind: will I be able to do the new job? But I did start the new job full-time beginning of January. thanks. I was able to do it, and I performed just fine. And what I learned from this is kind of like disease and performance, in my case and my perspective, that I might have some direct negative effects, like being tired or needing to go to hospital or so on. Actually, I don't have too much of that, but at the beginning especially I had. But then there are some indirect positive effects, which actually, which I would have hope I would have known, and which I have seen in some other patients as well. That kind of, it led to strong feeling of survivorship and courage. Courage and survivorship led to purposefulness and also courage, and this courage and purposefulness affect performance a lot. So courage to be honest, courage to take action, and purposefulness. So whatever I do at work, I'm thinking that, is this how I want to be using my time? Does it serve the purpose of my work? And so on. Then third section. So relevant data. My patient and statistician perspectives on few topics. So estimates for treatment A. What was in the label for treatment A? Nothing for my disease. So it is an off-label treatment, and then it is kind of going in a fog and with a Google and so on. What would have been relevant for me as a patient? What is the actual risk of a given side effect? What is my efficacy if I can tolerate the treatment? And are there ways to deal with the side effects? An estimate for 1 and 2 is principal stratum. Then a little bit about type 1 error and type 1 error control and multiplicity. So I do think type 1 error control for primary endpoint and pre-specification of secondary endpoint is really important. I do think that as a statistician and as a patient. However, my time as a statistician, I would rather use on collecting data reliably than optimizing and changing testing hierarchy and alpha splitting. And at the end, it might still be that I kind of select, I mean, of course, with the team, but so on. And so as a patient, I like to have the relevant secondary endpoints in the label. And unadjusted p-values are more informative than adjusted. And then a little bit about advanced statistical methods. So there you see the first draft for this presentation. And patient journeys are complicated. And it is good to look at the total picture and try to understand all relevant dependencies. However, answering the important sub-questions reliably is more important than being able to model everything simultaneously. And here you see what were the sub-questions for me, which I tried to answer here as a patient. And then one important question which still remains: does the treatment A impact the cancer as well? And summary. So data is powerful on its own. And this was really like, as a statistician, I realized that how much it can impact someone who is reading that data. Then beyond treatment, we can impact patients and colleagues' lives. And the last and the most important is that the patient mind is unique. So my order was that, someone else's order is something different. And I can only speak on my own behalf, not for anyone else. And then relevant and reliable data is crucial to support those choices. And where am I now? So complete hematological response since 2019. And I am weekly low dose injection of treatment A. And I do get some mental health support for other part of life and work. And I am living a full and happy life. And with this one, I want to conclude my presentation and thank you.
Thank you. Right, thank you. Thank you. We'll now have a panel discussion. We've had 4 very different perspectives on patient preferences, and we'll see if we can somehow get them together. I think it's safe to say that all stakeholders are interested in patient preferences and would like that to be included in the label. We also have a second patient for the panel debate. It's Johan Hellsten. He's a Master of Science in Neurobiology, and he's a PhD in molecular psychiatry from Lund Universitet in Sweden. He's been with Lundbeck for 15 years and since 2009 in various roles in the R&D organization. Almost 9 years ago, at the age of 46, he was diagnosed with early onset Parkinson's disease. In 2020, he joined the newly formed Global Patient Insight team at Lundbeck as a senior specialist, working primarily on the company's PD pipeline. And before I give the floor to you to ask questions, I would ask Johan to make a statement first.
So, first thing to say, I'm by no means a statistician, so that's important to bear with me. Also, I woke up at 5:20 this morning, wondering what I was going to talk about this fine day. And I started out by trying to sort of come up with something that would sort of resonate with the overall theme of this session, the patient preference studies. And this is, after all, the country where we have an anti-PowerPoint party, I've learned. I have no slides, so bear with me if it becomes sort of incoherent or rambling. As we said, I was diagnosed with early onset Parkinson's disease in 2016 and started treatment. I don't know, are we allowed to say brand names, drug names? I don't know. It was a drug that was marketed by my current employer, so it was sort of strange start to this disease journey. The mainstay in treatment of Parkinson's disease is levodopa, which was introduced in 1968. And after a few years of that, patients normally start developing this wearing off phenomenon where the drugs don't work really. So coming in an off state, as it's called. And what you do then is you can add, for example, dopamine agonists, or you can add something called COMT inhibitors, which sort of prolong the period where the amount of levodopa that is available to the body. And I was pondering the options there, discussing with my neurologist, and I read about what was available. And there was a second generation COMT inhibitor, but it had the quite unpleasant adverse event profile of, first of all, diarrhea. It comes sort of out of nowhere after months or even years of treatment. And then to sort of add to the stigma of having Parkinson's, and sometimes really, I mean, I feel like I'm 200 years old and trying to walk around. To add to that, this treatment gives you discoloration of the urine, becomes dark brown, but not only of the urine, but also the saliva and sweat. So not really very pleasant. But at the time, there was, in fact, an option to this, a third generation COMT inhibitor that was approved by EMA in 2016. Thing is, this was not available in Sweden. It was not marketed there. It was, I think, the company marketed in Germany, UK, France, Spain, Italy, and so on. I don't know why. And my neurologist was very knowledgeable, couldn't for his life understand why they not pursuing this. And I was trying to get hold of this, which I finally did. And after a few years of sort of getting it from a online pharmacy in the Netherlands, it became available in Denmark, where my employer, I work in Denmark, so I have access to medication from the Danish system. 2 years after that, was a discrete choice experiment that was published, showing that what was described as in the label for the first COMT inhibitor, the one with the really nasty side effects, described as harmless, meaning that it's not medically important, was in fact the sort of main deterrent from patients choosing this treatment. So when they were faced with the either having a 40% risk of discoloration of saliva, urine, sweat, you would need an extra hour of on time to sort of ameliorate this. And then a 10% risk of diarrhea would require 40 minutes more of good on time. And then the once daily dosing, that is new treatment provided in contrast to the old one, which you had to take every time you took a tablet of levodopa, which I currently do 8 times a day, was also considered important. So this kind of information, I think, would have been interesting to have in the label somehow. I don't think it was something that a company would have thought about in 2016 when the drug was originally approved. But definitely now it's available out there. Yeah, I think that was it for now. And I think other people will be able to take questions.
I'll just make a point and then a quick question. Can I point out that the UK used a patient preference study, multi-criteria decision analysis in 2019 on product Evenity to overturn a heavily negative CHMP decision. So it has happened before. Sorry to burst the bubble of the first speaker on that one. We got there first. And is it Muna? Yeah. So my understanding is that this was all driven by the FDA asking their statisticians to write a report, and they emphasized adherence. And you wrote an article on adherence and how to use it. You called it the tripartite approach. You wrote it with 2 other authors. And I was rather hoping you would talk about that in this, because it brings forward the patient perspective. Okay, and so I wondered, have you let that go, or are you still pursuing it? Or go on, tell me what you're doing and how you're fitting it into this magical world of intercurrent events. Thank you.
Well, first of all, thank you for giving me that. How do you call that when you give me that opportunity to talk about it? No, so I haven't. Of course, I have not forgotten about the tripartite framework. I think you captured it a bit in some of your questions. Indirectly that you put on your slide. I think, yeah, I mean, I'm still very much of the opinion that there are additional questions that are relevant to patients. And in that paper on the tripartite approach, we've done a couple of interviews with clinicians, patients, but also ourselves, let's say, and we try to categorize the questions that most people may have into 3 categories that correspond to 3 estimates, so the tripartite approach. I think the challenge with that a bit is we are sort of making out of 1 question, we are creating 3 questions, which are all very relevant. But I think for regulatory decision-making, we still need 1 gatekeeper, let's say. 1 question to open the door that we can have the conversation, which can be like a utility or something. That is why I said it would be important to distinguish more clearly between testing and estimation. So let's have 1 gatekeeper question. But once we pass that question, I would be very happy if we could look at more detailed questions like those laid out in the tripartite approach.
So it's just a comment, okay? I use your scheme to ask questions to companies. So I would encourage you.
To use it myself.
You've caught my imagination, yeah.
I will
So
talk
I
to my
would encourage you
manager
to
and some other
pursue it,
people
yeah.
here. No, thank you.
Thanks for the inspiring session. I wonder whether you could comment on, I think it's to all of you, on the relevance from your perspective and reliability of patient-reported outcomes. And then it seems to be a bit kind of an imbalance between these 2 worlds. The patient-reported outcomes, where we collect sometimes 4 or 5 questionnaires in a trial, and patient preference studies, where then we have no budget anymore left for them to conduct in a trial. Any comments on this?
Thank you for that question. Actually, it's a common question I get all the time. The distinction between patient-reported outcomes and patient preference is that patient-reported outcomes are realized outcomes. They are statements of what someone is actually experiencing, what is going on. Preferences are by nature ex ante, which is given a choice, what would I do? So they measure totally different concepts where a PRO could say, I can do X. A patient preference study would be able to tell me how is it important is it for me to be able to do X. So, they're very different things. From a regulatory perspective, patient-reported outcomes can have the same characteristics of a good clinical endpoint. And so, and have the reliability and all of those other characteristics that we would look for in endpoints. So they are treated differently because they're treated as outcomes or potentially as endpoints, whereas preferences are really meant to characterize the rest of the results, where preferences themselves are, with few exception, not endpoints in and of themselves or outcomes in and of themselves. They really provide the weights, essentially, that for what Francesco was talking about previously. Could even provide weights for PROs, for that matter, although the PRO people don't like me to say that very much. But yeah, they serve different purposes. They measure different things. Did that answer the question?
Yeah.
Right.
Well, the first part is actually whether they are relevant. I understand they can be relevant, but are PROs relevant and maybe a patient?
Oh, you mean in terms of the information they provide? Yeah, I'd be curious to know what the patients would say about that, because I don't know.
I think the short answer is yes, but we need to stop thinking that there is a universal categorization of endpoints between relevant and not relevant. The relevance is in the eye of the beholder, and we have to learn to deal with that and to measure that systematically.
Yeah, so maybe I add to that systematically kind of thinking that was a bit so I know it's very difficult to get PRO data into the SMPC at the moment in Europe, but that was kind of a little bit, I didn't use PRO data as any of the examples, is that when we then maybe send a comments back and take it out of the SMPC and maybe have there some justification why we did that, the response that comes back, it's important for the patients and it's put back in. Whereas our comments, well, I mean, I can speak for the whole network, are usually actually related to that it's not reliable. So I would really urge to look why we asked to take it out. And then if you don't agree with those, answer with those points about the reliability. So why should we trust those results? And then also, of course, plan from the beginning to do it well, because that helps you already like 80% there, right, on the way. So I think that's kind of was in a way the message behind my talk, right? That then look at the question, look at really what we said and answer to those both, and don't just say it's important for the patients, because that's only like one part of what needs to be there to get it into the SMPC. Anna, please.
Yeah. Yeah, I think they are. Then I would say that well, first of all, I'm not so familiar with those, but I would say that well, I have worked as a statistician in some trials, and if I see that there are some sub-questions, then actually the sub-questions might be kind of, I would think as a patient, they might be more interesting that if I know that I have some symptom that does it ease that symptom, which is, I'm thinking this, I was working on rheumatic diseases, and there you have something that can I put my clothes on easily. So then instead of having one score, I think it might be that the sub-question, you know.
Yeah, I know.
Yeah. Yeah.
I also have a question on patient survey and especially methodology on risk assessment. If I understood correctly, the information you should provide to the patient on, for example, cancer risk or other adverse event is based on the frequency of this event observed in the study population. But it's not the same as the probability of having this event for the particular patient, because I assume you don't take into account genetics, comorbidity. So what information is provided, basically, it's what is the occurrence of this event generally, but it's not my risk as a patient of having this event, because I might have different profile in terms of genetics, family history than another patient. And that would change my tolerance to the risk versus benefit. How do you take that into account?
So these are sample level estimates, as you pointed out. So we do the means across the sample and maybe by subgroup. Our first goal is to ensure that we describe the attribute, each attribute, in a way that is clinically accurate, but also understandable and conveys the full impact of whatever event might occur, whether that's a benefit or a risk. There are ways that we could, in theory, adjust for all individual characteristics. So, and risk factors. Unfortunately, it becomes too complicated, and then the survey becomes really long. So that's the challenge. So often what we do instead of adjusting in the experiment itself for those baseline characteristics or those risk factors, is we will elicit that information that might help us understand who might be at higher risk and who might be at lower risk, and see whether those who are objectively at higher risk have preferences that differ from those who are lower risk. Now, there are lots of assumptions behind that. So it's a very much that needs to be interpreted qualitatively. But this is, if we were going to do what you were suggesting, we would need to use a method different than the DCE, where we were getting actually your specific individual level estimate of your risk tolerance. Then I can adjust for it easily. But the DCEs, because we look at a pattern of responses, I need a sample to estimate the data. I can't do it on an individual. We don't get individual level estimates. So it's very difficult to implement. It's probably not impossible. It's just extremely difficult.
Yeah, thank you. I was just wondering if patient understand that actually it's not their particular risk. Does that explain to the patient?
We have to make it very clear that yes, in fact, that is the case, that what we are saying is not, this is something you will experience or you will face. It is that the evidence for this product shows. So we treat it as if a patient is looking at a label, and the information in the label, which comes from a clinical study that isn't them, but patients in that study experienced, et cetera, et cetera. We have to be very clear about how we communicate that.
Brett, can I just add something to your excellent answers, also to the previous one? Again, the I imagine the cost of the study depends on the purpose. And there are many types of study designs, if they're not used for, let's say, an inferential purpose, but just for a supportive orientation, let's call it more of a secondary purpose, that could probably take a very different format and resource than a fully blown DCE with a huge sample size. In fact, in our world, we find that instead of just experts voicing opinions, having a focus group and recording, eliciting preferences in a sort of decision conference setting is already much more informative than, let's say, the voices of individual experts. So it's very important to see what you want to use them for and adapt. And the last thing that we want is that we create another monster where for unclear regulatory ideals, every study has to be of the highest, disproportionate highest standards. No, the standards have to be proportionate to the objective. And the other thing, what you touch on is also a critical question, which kind of pervades all of the benefit risk assessment, which is, are you doing this from a population perspective to say how much you value a population that increases their survival by a median of 1 month? How much you value that? And you can ask that to patients, and it's very informative to know what they think about that. But it's an entirely different question than asking them, would you take this drug with these risks and this probability of success? Decision under certainty on one side, decision under uncertainty on the other. And these are very different questions that mimic a little bit very different regulatory decisions of the full approval and with, let's say, more of a group effect in mind, and the conditional or accelerated approval where you have lots of certainties and you really want to know, are these risks and expectations compatible with what patients find useful? So critically important question.
I already kind of lost about the questions, but there is one point which I would actually also like to do that. I mean, when I told my patient story, I mean, I wanted to know everything. then really also about what do patients want to know? There is always a difference, and this is something what I discussed before this presentation with one medical doctor, that it might be also that there are patients who just want to have, you know, that the doctor decides, and that's also totally okay. And yeah, and then the doctor perhaps makes it in a different way, and so on. But
yeah. So one final question.
In fact, I had a question, but I was just communicated the results of an informal audience preference study that says that between the benefit of continuing such a fascinating discussion and the risk of delaying lunch further, I was told that the trade-off is very difficult. So I'm afraid I don't.